Are
Okay.
oh crap. How's it going?
Good, good. Feeling better since yesterday, so I've been trying to get back into the grind. Um, yeah, I really wasn't able to take the full end because it was like Saturday and I was already feeling pretty bad and I didn't really feel
Sure,
the
sure.
right. But I'll definitely take one. I'm gonna take one for sure. The Blueprint 10th one on Friday. Um, but yeah, I got a lot of, I actually got a lot of questions today, so hopefully we can get
Okay.
all of them. Let me share my screen. I wasn't gonna go through some couple passages, but I think first we can kind of go through these like little discrete questions. Um, yeah, I've been trying to do a lot of the fundamental questions on the Jack Weston. Uh, they've been really helpful actually.
Yep.
Yeah, so I'm a little confused with this, but yeah, I'll let you read it, I guess.
I read it.
Oh, okay. So, um, I'm kind of confused here with why it's all of them because a sphingolipid does not have an ester linkage, right? I don't really understand why saponification would work for all of them.
Okay. Well, what can you tell me about um a sphingolipid?
Okay, I got you. You know how it does this. Okay. Um, yeah, so it's got like a something like this, right? It has like a OH and an OH, then long chain. I don't really know how many carbons, but and then I think it's something like a nitrogen here. I think it's NH2, I think. Um, yeah, typically a fatty acid would form here, but it would be connected by an amide linkage, right?
Hmm?
So I'm like, if saponification is strictly for typically ester linkages, then how would it break a amide? I mean, yeah, how would it break this?
Okay. Let's see. Well, a phosphor okay, so let's talk. All right, so what's saponification?
Yeah, saponification is using a strong base to break typically a triacylglycerol into the fatty acid salts and the um yeah, the fatty acid salts into the glycerol backbone or whatever backbone it is. Typically it's a triacylglycerol, I see.
Yeah. Um, so let's see.
It seems that it also works for phospholipids, but it's apparently really slow. I tried to check online, but online keeps saying that sphingolipids apparently it doesn't work. So I'm a little confused why it's the answer.
Got it. So okay, so what can you so how about let's look at the phospholipid first. The
Okay.
phospholipid should be something with, fatty acids connected to via an ester linkage, right?
Yes.
So, okay. So I guess, well, there isn't a choice well, there isn't a choice, I guess, that says one and three, but it's
Yeah.
that it shouldn't be just one, at least knowing about phospholipids, right?
Yeah, sure.
Um, now when you saponify something, what happens?
Yeah, typically you use something like a strong base and you break it into the fatty acid salts
Okay.
and the backbone, typically whatever backbone it is. It seems like it's a way that a lot of soap molecules are created. So it creates like these like micelles, I think.
Okay. So I think, let's see. I think they may have messed up the phrasing of this, but let me. see. Um, okay. So, okay. So you talked about a um what's that? Okay. Um,
Yep.
so sphingolipids can include um things like, let's see. Sorry. Um, hmm. Okay. Okay, why don't you look up sphingomyelin?
Okay. See. Single myelin. Okay.
So, what do you notice any? So, okay, so this question is tricky because they're talking about an ester linkage, but that could include a phosphoester linkage.
Phosphoester. Like this?
Yeah.
Hmm.
So I think
Okay.
the questions worded pretty poorly. Um, so
Phosphoester. Okay. Dang,
yeah.
yeah, this is so specific.
Well, yeah, I think the question's worded poorly because it says fatty acids connected via an ester linkage,
Okay.
right? And even in the example of a sphingomyelin, it's not a fatty acid that's connected.
Oh, sorry, my bad.
Oh yeah. Yeah, even in the sphingomyelin, it's not the fatty acid that's connected via an ester linkage. It's the phosphate group.
Yeah,
So yeah, so this isn't like the best question. I think that it would make sense to choose Roman numerals one and what was it, three?
phospholipid and
Uh-huh.
triglycerol.
Yeah. So yeah,
Okay,
let's go back to the question.
sure.
So okay. So yeah, this isn't the best worded question. I'm totally with you on that, but I think the logic that would have been useful here is like, okay, triacylglycerols, we know for fact that that, you know, would have fatty acids connected via an ester linkage, right?
Yeah.
And phospholipids should have that as well, right? Because a phospholipid is just like a triacylglycerol, except you remove one. Instead of having three fatty acids, you have two fatty acids and one phosphate, right?
Yeah.
And so phospholipids still have fatty acids, two fatty acids connected via an ester linkage, right?
Mm-hmm.
So okay. So that means okay, Roman numeral one and two, two and three, one, two, and three. So we know that one and three must be true, right?
Yeah.
So I think the logic would have like should lead you to D because that's the only one that includes Roman numeral one and three,
Okay.
right? Um, and but I'm with you on the sphingolipid part because yeah, as you said, we're not having. I mean, I gave you an example where there could be an ester linkage of sphingolipids, but that's
Mm.
a phosphoester,
Yeah.
which is not an ester that's linked to the fatty acid. So I think that this isn't a really good question because of that. If
Okay.
they did not include this parentheses, saponification of lipids include, I think that would be a lot better.
Okay.
But I'm totally with you on this. In that, yeah, sphingolipids typically, I mean, they don't really, yeah, I can't really think of an example where they have ester linkages. I mean, there's ester linkages to the fatty acid because
Okay.
the sphingolipids, you know, can have an, I guess also we want to understand that a sphingolipid includes quite a few types of sphingolipids,
Yep.
like a phospholipid, a glycosphingolipid, right?
Mm-hmm.
So,
Mm-hmm.
um, also, let me take a closer look at glycosphingolipid. So, the glycosphingolipid has acyl chains that are attached to hydroxyl groups. And if you, let's try this.
It says glucose chains, chains of, oh, any sugar, I guess.
Okay. Hmm. Yeah. Yeah. I'm with you on that. Yeah. I don't think the question is worded properly.
Okay, sounds good. Also speaking of phosphoesters, um, so yeah, I just wanted to go over phosphoesters real quick because you just mentioned it, and it's been a while since I looked at it. I remember sometimes uh that always messed me up. So essentially, phosphoesters can act. They're like basically the same thing as an ester linkage. The only thing is it's just a phosphate group instead of a carbon, right? That's really the main thing, right?
Hmm.
Okay, sounds good.
Yeah. So this question, I think it's the stuff that they wrote in the parentheses about the fatty acid that makes it wrong. Because
Hmm.
even in the explanation, they're just asking like which of the three lipids are saponifiable, right?
Yeah,
It doesn't say anything about whether they have esters or not in the solution. So I think that it's just bad wording on their part. They probably, mean so they mean to ask which of the following lipids can be saponified, and I think they're saying they're thinking that if you may not remember what saponification means, that they're telling you in the parentheses by saying that the saponifiable lipids are ones that have fatty acids connected via an ester linkage, but
yeah.
that's not something that we see in sphingolipids. Like even in the example I gave with a phospho ester,
Mhm.
that's still not because they're talking about fatty acids connected by an ester linkage, and that's not true. So
Okay,
yeah,
okay. But overall, the saponification does concern with only removing the fatty acids, right? Like for sure.
yeah. Generally, that means that you are breaking esters
Hmm.
down, right?
Yep.
Through the addition of like a strong base, and then you can get um, the component fatty acids essentially.
Okay, sounds good.
Yeah.
We'll work our way back from this. These are just more questions.
Okay.
Yeah, I kind
So
of,
yeah.
but there's like a part of it that I just, yeah, yeah.
By the way, I want to make so when did I have you ever do like the Jack Weston content diagnostic?
Yeah, we did.
Okay, okay.
These are just extra problems that I'm just doing. Because last time you told me, and I'm like,
Yep.
I realized I just haven't gone as deep. But like now, I'm like, I
Mm-hmm.
might as well now not on March 8th, right? So
Got it. So let's see. So the one, all right, the on the right we have because like there was like a manometer problem in the content diagnostic that we
yeah, I
put.
remember that one. I remember that one.
Hmm.
But I didn't. But like for this, what
Yep.
threw me off was this part.
Yeah, yeah, yeah.
It's
Okay.
like I don't really know.
All right,
Yeah.
let me take a look. So the part on the right is open to the atmosphere, and from the fact that the liquid level is higher on the right, we know that the pressure from the left is going to be larger than the atmospheric pressure, right?
Yep.
Okay. Um, so and then we have gas X, P1, V1. The two V's are equal, and then we have some stopper in between the M, and then gas Y, P2, V2. And they're saying when M is removed, the mercury levels on the right and left side of the tube become equal. Okay. Which of the following is true regarding P1, P2, and PATM? Okay. So if opening this stopgap here causes the pressure on the left and right side to become equal, that means the pressure on the left side would decrease, right?
Yeah. If it was to equal, yeah.
Okay. So we should have, oh, this is weirdly written with like less than, greater than, wait. Oh, okay. Less than, greater than. Okay. So we know that P2 must be greater than PATM.
Yeah. I'm good with
That
that.
makes sense?
For sure.
And if we open this stopcock here, the overall pressure on the left decreases, right? And
Mm-hmm.
and that's when you mix gas X with gas Y. So gas X must have a lower pressure than P2 then. Because if it had a higher pressure, well, if it had the same pressure, you should not see any change in the levels here of the mercury liquid.
Okay. And if it was higher, this thing would be even up.
Correct.
But
But
the fact that it's lower, okay.
since they're saying that when you open this, the level goes down, it means the overall pressure on the left goes down.
Oh, okay, okay.
Yep.
See what it's saying.
Hmm.
And don't assume. Put together. Okay. So again, so because of the fact that we know that pressure two is greater, but the second they take it out,
Yeah, the second they start
levels are
heating it with gas X, it goes down the mercury level, which means that the pressure on the left it must be decreasing.
right. Because if it was again, if it was equal to or increasing, it would stay the same, or the liquid go even more up. Okay. Sounds
Correct.
cool.
Yep.
Okay. I think we can do 44. Okay. Yeah, this one I didn't really understand about real gases.
Ah, I see. So what
Yeah.
is the difference between ideal gas and real gas?
Yeah, so typically ideal gases are essentially when you're at really high temperatures and low pressures, because that allows you to make like certain assumptions, I guess, like, intermolecular forces are,
So what you're saying is conditions in which an ideal gas may behave like, or rather, you're talking about conditions in which a real gas would behave as an ideal gas, but what is the actual difference between a real gas and an ideal gas?
I guess when a real gas like deviates from those conditions, right? No?
Well, yeah, but what are those conditions?
Oh, I mean, typically for real gases, it would be when it's like really high pressures and like low temperatures.
Oh no, so you're giving me examples of how of situations in which a real gas behaves like an ideal gas, but what like what actually makes an ideal gas an ideal gas? There's two main factors.
Two main factors.
Yeah.
Um, if there's like no like volume is insignificant.
So
Something.
so so okay. So basically the volume if the so the volume of the actual gas particles
Uh-huh.
is negligible
Yeah.
in an ideal gas, and the intramolecular forces between the molecules of the gas are also negligible. They're basically zero.
Okay.
So
Yeah.
that's what makes an ideal gas an ideal gas. Now, what you said was at high temperatures and low pressure, I think, right?
For yeah, for ideal gases, yeah.
So that would be for a real gas to behave at like an ideal gas, right?
Oh, okay, sure. Yeah.
Like here, I'll write this. So
I didn't think about it that way, but yeah, that actually kind of makes sense.
So an ideal gas is when the volume of the gas particles themselves are negligible, as well as the intermolecular forces between the gas particles, which are also negligible. Okay? Now,
Okay.
if you take a real gas and you put it in an environment in which there's high temperature and low pressure, that means that, so the high temperature means that the molecules are moving quickly, right?
Mm-hmm.
And if they move quickly, they don't have a chance to sit around and interact with one another, right?
Okay. Yeah.
So you could take a real gas and have it behave like an ideal gas at high temperatures. And
Mm.
also, if there's low pressure, they're also not going to be near each other, having, intermolecular forces acting upon one another. And also if it's a low pressure, you'll have a high volume because you'll have the molecules really spread apart. And that would make sense because if you're considering the volume of the gas negligible, then that would make sense in a low pressure environment, right?
Mm-hmm.
Okay. So, all right, now they're saying this graph here shows the ideal gases at a temperature of 273 Kelvin. So we see the pressure, y-axis, the volume, v-axis, and this is an inverse relationship, right?
Yep.
Okay. Now they're asking what would a real gas look like? So I think what you chose, let's see, at higher pressure, volume would be smaller. At higher, wait, oh, the difference for real gases. So I guess they're saying the first two things are for an ideal gas, I guess. And this is kind of weird worded weirdly too, but okay. At higher P, oh, this is just saying for real gases alone. Okay, so at higher P, V would be smaller. At higher V, E would be smaller. Okay, compared to at higher P, V would be larger. At higher V, um, P would be smaller. Okay, so this is worded pretty poorly, but they're asking what conditions would this ideal gas behave like a real gas, right? Um, oh, and then here in the explanation, they actually tell us what a real gas is. So you can see it says the opposite of what I wrote for an ideal gas, right? We have
Yeah.
non-negligible volume and experiencing intermolecular forces, right?
Mm-hmm.
So, and this would happen at higher pressure and lower temperature, right?
Yeah.
Okay. And you're saying the pressure versus volume graph shows the deviation of real gases from ideal gas behavior. The plot of pressure versus volume should ideally align with both experimental data representing real gas behavior and theoretical data. Oh, can you go down a bit?
Yeah, sure.
Theoretical data calculated using Boyle's law. Got it. Um, However, observations show that at high pressures, the measured volume of the gas exceeds the calculated volume due to intermolecular forces. Okay, so let me. Okay, I'm gonna share something with you.
Mm.
So, okay, it said that at high pressures, the measured volume of the gas exceeds the calculated volume due to the intermolecular forces. On the other hand, at low pressures, the measured volume of the gas remains below the calculated volume due to the interactions between the molecules. So you're going to have a pressure, let's see. Okay, so in a real gas, the intermolecular forces
Mhm.
mean that the particles of the gas will collide, or the intermolecular forces could be causing the particles to attract one another.
Mhm.
And if they attract one another, there will be more, you know, collisions between the molecules rather than collisions between the molecules and the walls of the container.
Okay. They're colliding with each other compared to okay.
Yeah. Now, when particles of like a gas collide with the walls of a container, we can measure that as pressure. We could say pressure of something is, pressure of something inside of a container is essentially the molecules of that thing pushing up against the container walls, right?
Mhm.
So, um, so we should see in a real gas a smaller pressure.
Smaller pressure. I mean,
Let's say that this is a container and these are two molecules of the gas. And this one's moving to the right this way, this one's moving to the left this way. Now, this is going to be an ideal situation at which there's no intermolecular attraction between these molecules. So that means
okay.
this one is going to hit this and like maybe bounce off, come here, hit that, bounce off. This is going to hit here, bounce off, come here, hit that, bounce off. You have a lot of these uh, particles hitting the walls of the container, contributing to a high pressure, right?
Isn't isn't pressure low in an ideal gas?
No, the pressure would be higher, but I think what you're thinking here is, um, again, like conditions at which an ideal gas will behave, or conditions at which a real gas will behave as an ideal gas compared to conditions in which a real gas behaves as an ideal gas,
Okay.
right? Um, so in this ideal gas, you could see that these particles that I drew are not hitting each other, but they are hitting the walls of the container, which means a higher pressure. Okay. Now, in a real gas, for this one moving this way, this one moving this way, they are going to have some attraction between one another. And if they are attracted to one another, they're going to be closer to one another rather than passing each particle by and hitting the walls of the container, right?
Yeah, they'll be hitting it less. Okay.
So this would be a low pressure in for an ideal for sorry, a lower pressure for a real gas compared to an ideal gas,
Hmm.
right? Because I mean, there's like so like the equation here is going to be pressure of an ideal gas is pressure of the real gas plus this term here. And it comes, so this A term is something called an attraction coefficient, meaning that this um, attraction coefficient can be added to the pressure of a real gas to give you ultimately your ideal gas. But we do see that the ideal gas pressure is greater than that um, of a real gas. Okay.
Okay.
So, okay. Now, for the volume part, um, the in an ideal gas, you will have uh, you'll treat the actual volume of the particles as negligible, which means that, like, so for this drawing over here, for this drawing over here, like,
Okay.
I'll use like purple here. So, like, this, like, volume of these particles are not expressed in the actual volume of this container because we treat the actual volume of the particles as zero.
Okay.
But for a real gas, we don't treat the volume of the particles as zero, so they have their own volume. And if they have their own volume, the overall volume, right, would be um larger, right?
Want to be larger? Yeah.
So we can say like low volume here, high volume there, right?
Sure.
So here they're telling us that um, high pressures, the measured volume of the gas exceeds the calculated volume due to these intermolecular forces. And then on the other hand, at low pressures, the measured volume of the gas remains below the calculated volume due to the interactions between the molecules, the attraction forces. So what this would mean is that at higher pressures, you would have a larger volume because, and all of these are talking about for a real gas, by the way, right?
Yeah.
So, yeah, so we know that. So, okay, at higher pressure, the volume would be larger, which is what we see here. And then they say at higher volumes, the pressure would be smaller, which is what we see here. So they're really just asking us what would happen, like in the example that they gave for an ideal gas, which are these conditions, how they would, deviate as real gases.
Okay. Um,
So, okay, so I'm gonna write one more thing. So real gases behave as ideal gases at high temperatures and low pressures. Because that means you're going to minimize your intermolecular forces and you're going to minimize um, the uh, volume of the particles themselves. And then ideal gases can behave as real gases at low temperatures. Um, wait, hold on. Ideal gases as real gases. Yeah, the opposite. At high pressure and low temperatures. Yep.
okay.
So at higher pressures, volume would be larger because you consider the volume of the actual particles. Oh yeah, and then like the kind of like formula for that would be um, volume ideal is equal to volume of the container minus NB. And this B is just this thing called like bigness coefficient.
Okay. Um, kind of get it, but I don't really, I still don't really understand this part. Like, A, how you kind of went back to this, still explained it, but
Sorry, which part?
um,
So I'll
also
start
you're
with that.
saying also here you're saying the volume is real is greater than the ideal volume, and then the pressure of the real is less than the pressure of the ideal.
Yeah.
Okay. Um, yeah, I still don't get this part. How you said this
Hmm.
three, that's the answer. Like for some reason it just doesn't I still don't get it. I don't
Hmm.
know why.
Uh-huh. Yep. So at higher pressure, so they're asking us about what would happen if we had real gases, right?
We had real gases. Okay, yes. If we had real gases, yes.
Yep. And so we know that real gases take into consideration the volume of the particles themselves.
Mm-hmm.
Okay. So that would be like if so see this uh jar of water.
Mm-hmm.
So what if I did not consider the volume of the particles of water within this, the overall volume of this jar will just be the volume of the jar,
Yeah, okay.
as if it's empty.
And that would be like ideal gas, right? Like if
Correct.
typically, okay.
Now, real gas would mean I take into consideration the volume of the actual water molecules, which means that the volume of this would be considerably larger, right?
Hmm.
Because an ideal gas would be like treating it as if you don't even have like the molecules, like you treat the molecules as if they don't have any volume,
Yeah.
right? But if so, that would be like taking this and then treating it as an empty container.
Hmm.
So that's why your volume of a real gas will be larger.
Okay.
So volume is smaller, we can eliminate those, right?
Right, at high pressures, okay. Volume would be smaller, okay.
Yeah, the high pressure part like is not the most important thing here, but it does kind of just tell you like I guess like what I wrote here, like high pressure or whatever. But yeah, we're mostly just looking for what would make something or what would a real gas,
Mm.
how the real gas will be different from an ideal gas. Yeah. So, and then the second part at higher volume, pressure would be smaller. And we know that since the pressure is thought of as the particles hitting the walls of the container, if you had a and in an ideal gas, we don't have any of the particles, being attracted to one another. So every particle will have to hit the walls of the container.
Mm.
But in a real gas, the particles can attract one another. And if they attract one another, they're going to basically stick together rather than hitting the walls of the container, right?
Right.
And that means that the pressure would be smaller in a real gas because you will have the molecules attract one another rather than always hitting the walls of the container.
Okay, that's making sense now. Okay. That's
Yeah.
making a lot more sense. Okay. Yeah,
Hmm.
I think the thing that keeps throwing me off is the part that it says higher pressure, but I see the I see the volume would be larger in that other part.
Yeah, yeah. This is, the way that they presented this question, like if you scroll up, can kind of make things a little bit confusing because first they just show you like, this diagram here. Um, and then they talk about at higher pressure, at higher volume, etc. So I think that can make it tricky, but the reason that they're showing you this is because we know that there's going to be an inverse proportion between pressure and volume, right?
Hmm.
And that looks like this shape, and they're just and that's according to the formula, right? Like, or rather the law of, I guess, Boyle's law, which is the
Yeah.
inverse proportion between volume and pressure. So,
Hmm.
so in an ideal gas, the reason we like to use ideal gases is because they fit with the law perfectly, right?
Yeah.
But you can see now for the drawing over here, you could see that the difference between the ideal gas and the real gas is really apparent at the higher pressures. Because at the lower pressures, like down here, they kind of intersect. There isn't much of a difference. It's hard to see the difference at regular pressure and volume. But you can see that this difference is greater between ideal and real at high pressures and at high volumes.
Mm.
That's
Okay.
where they really show that they're really different. So that's why the question choices are saying at higher P, at higher V, right?
Mm, okay.
Because, it's pretty much what we wrote here. Like, so, like ideal gases behave as real gases at low temperatures and high pressure. I guess, high pressure could also mean, um, low, low volume and stuff.
Okay. Sounds good.
Yep.
Actually, I don't think I got that top part. Oops. Okay. Cool. Okay. Let's see what else we got. Where was it? I'll try to do that passage. Actually, wait. I think let's do this one instead. If someone can come back to that one. Um, yeah, I think it was hopefully making me go over this.
Okay, for the process to occur in cells, the protein folding must be a spontaneous reaction at physiological temperatures. Consider a straight polypeptide chain in water. As the protein folds to form its tertiary structure, the surrounding water decreases in temperature. Okay. The change in free energy, enthalpy, and entropy relative, um, respectively, of the environment, water, and protein are. So, okay, our change in free energy is our Gibbs, right?
Yeah.
Enthalpy is our delta H, right?
Mm-hmm.
Entropy is our S.
Yep.
And they're telling us that the surrounding water decreases in temperature. So our delta T would be negative, right?
Mm-hmm.
Okay. So, oh, and yeah. So they said that it must be spontaneous, which means our delta G would be negative, right?
Mm-hmm.
So we can get rid of A and B, right?
Yep.
Okay. Now we want to figure out what the enthalpy and entropy value would be. So we can take our delta G is equal to delta H minus T delta S, right?
Yeah.
Okay. So we can have, so if it's losing, sorry, if its temperature is decreasing, we can say that our, wait, let's see.
There's decreasing.
Yeah, if the temperature of like the surrounding water decreases, this would be an endothermic reaction, right?
The temperature is decreasing. Yeah, because there's no heat being released, heat is being absorbed.
So that means our delta H must be positive, right?
Oh, okay, okay. That makes sense.
So that's out.
Because the thing is, the reason why I was like really stuck and I kept pressing negative, because it said the protein was folding. So I'm like, I mean, I guess there's like interactions and like bonds and stuff happening. So that's why I assumed, or I was trying, I was stuck so much on like, how is it possible that it's positive? But I mean, I guess the temperature makes sense.
Well, so sorry, you're saying that when the protein is folding, what were you saying?
Yeah, isn't it as the protein folds, like wouldn't the enthalpy term be negative? Because you have a lot of like bonds forming to some degree.
Well, that so the enthalpy is a measure of, well, it's a measure of like, bond energy and stuff, but
Mhm.
ultimately it's there to tell you if something is going to be endothermic or exothermic,
Okay.
right?
Yeah.
But what does happen when a protein folds is the entropy term, because
Yeah.
before it before it folds, the entropy is going to be um negative because there's an entropic penalty before it folds. And so it will fold in order to reduce that entropic penalty, meaning an increase in entropy, meaning an positive delta S, right?
Sure.
So 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 yeah, that's why your entropy term, if you have an increase in entropy, right, it should be a positive term, right?
Sure. Okay.
So yeah. So that could be so yeah. So that's one way of answering this by like knowing like, conceptually what they mean by, enthalpy and entropy. But let's see if we can find it if we could do like the math here. So let's see. We have we have a negative T oh wait, I wrote this. Wait, T delta S. Okay. And they're saying that the temperature decreases. Okay. Okay. Um, well, yeah, it doesn't seem like because yeah, I wrote like the delta T is negative, but there's only a regular T here. But yeah, you would solve it like, by looking at the way things are worded. Yeah. So the surrounding water decreases in temperature. The reaction here is endothermic, meaning delta H is positive,
Yeah.
and me and oh and when it's folding, it's going to increase in entropy. So delta S is also going to be positive. And since they said it was spontaneous, we know that the Gibbs is negative. So
Okay.
that's pretty much it. Yep.
Um, what? So yeah, like that enthalpy, like so it's an endothermic or exothermic. I
Mm-hmm.
feel like that's something we can kind of it's still kind of get so it's should I be thinking about it as like when protein fold, would that be considered bond forming or no, right? That would not be considered any of that, right?
So okay, your delta H, which represents, that well, it could represent bond dissociation energy and stuff for covalent bonds, and that's more along the lines of if you're using like Hess's law, right?
Hmm.
But ultimately, delta H serves its function by telling you if something's exothermic or endothermic.
Okay. And typically it's typically more based on just the temperature, really, nothing really much else.
Well, of the surroundings, yeah,
Sorry.
because exothermic means you are releasing energy to the surroundings, and endothermic means you are taking energy, meaning heat energy, from the surroundings. If you give off heat energy to the surroundings go up in temperature.
Yeah.
If you are taking heat energy from the surroundings go down in temperature.
Okay. Sounds good.
Yep.
All right. If we have time, I guess we can come to that question, but I really wanted to go over this passage. Give me a tough time. Um, yeah.
Um,
Do you want me to read it or?
yeah, could do that.
All right. DNA damage from exogenous agents or natural processes can be repaired by homologous recombination. HR involves the breaking and rejoining of DNA strands using a homologous chromosome or sister chromatid as a template for the repair. Okay. Double-stranded DNA breaks are repaired primarily by HR or by a separate mechanism known as non-homologous end joining.
Wait,
NH.
sorry, is this blueprint fulling 10?
No, this is 9.
Okay, because I saw in your spreadsheet you wrote like 1228 for
I
blueprint
did.
fulling 10. So I'm wondering if you
Let me see.
not.
Where did I put my full lengths? Uh, I think I messed up somewhere. Hold on. Full length. Yeah, I think I was like I put that earlier, like early last week, because I was gonna take it. That was like the plan. But yeah, I didn't forget I was sick. So
Yeah.
where did it go? Yep. Yeah, so HR involves the breaking and rejoining of DNA strands using a homologous chromosome or sister chromatid as a template for the repair. Double-stranded DNA breaks are repaired primarily by HR or by a separate mechanism known as non-homologous end joining NHEJ. HR pathway proteins and mismatch repair proteins have been shown to co-localize in regions of DNA known as CPG sites where a cytosine is followed immediate by guanine. Methylated cytosine 5-MC is found to be in higher frequency in CPG dinucleotides, which cause an increased rate of mutation due to spontaneous oxidative deamination of 5-methylated cytosine. This deamination is 1,000 times more likely in single-stranded DNA than in double-stranded DNA. Okay.
So,
Um,
okay, let's talk about some of this stuff so far. So, saying that DNA damage from exogenous agents or natural processes can be repaired by homologous recombination, HR, and that involves the breaking and rejoining of DNA strands using a homologous chromosome or sister chromatid as a template for the repair. Double-stranded DNA breaks, DSBs, are repaired primarily by homologous recombination or by a separate mechanism known as non-homologous end joining, NHEJ. HR pathway proteins and mismatch repair proteins, MMRs, um, have been shown to co-localize um, in regions of DNA known as CPG sites. Okay. Where a cytosine is followed immediately by a guanine. Okay, so I guess that's what a CPG site is. Methylated cytosine is found with a higher frequency in CPG dinucleotides, which causes an increased rate of mutation due to spontaneous oxidative deamination of the methylated cytosine. Um, This deamination is 1,000 times more likely in single-stranded DNA than in double-stranded DNA. So maybe, oops, I'll write, let's see, CPG is proportional to 5-methyl cytosine, which is proportional to mutation rate. Um, Spontaneous deamination of the 5-methyl cytosine, and it's more likely in single-stranded DNA. Okay. Oh, and then HR-mediated deamination repair involves the 3' end resection and ssDNA formation. Scientists conducted an experiment to determine if this ssDNA processing figure 1 indirectly causes MMR recruitment to GT mismatches in double-stranded DNA. ssDNA processing is in figure 1, and they want to see if it indirectly causes MMR recruitment. Okay, so figure 1, homologous recombination repair pathway of DSB at CpG resulting in mismatch repair and chromosomal crossover. This asterisk next to the C indicates that it's the methylated cytosine. So, okay, let's see. We start off with, the double strand here with GC and CG, and it's the methylated Cs. A double strand forms, I guess. It's kind of, okay, so it's kind of weird. I thought like this was a double strand. Is it not a double strand? Is it only like this a double strand?
No, I feel like it might be this. I think this is just a double strand, right?
Okay.
Or maybe it could be both.
So, okay, let's see. Double strand formation. And okay, we have like a cut there. 3' resection deamination. Okay. Strand invasion, DNA synthesis. Okay, I guess whatever. That's just the recombination repair pathway. Okay. Experiment 1, DSBs in G1 arrested endothelial cells were induced by the introduction of SPO11, one bound SPO11 protein per DNA strand. Acts as a nuclease. Its active site containing a tyrosine residue that ligates DNA to promote break formation. Next, the cells were allowed to continue the cell cycle and were rearrested immediately prior to mitosis. Finally, the genome was sequenced using allele-specific PCR. Table 1 shows the position of de novo mutations in the molecule relative to the positions of CpG sites. Okay, so DSBs in G1 arrested endothelial cells induced by SPO11. One bound SPO11 protein per DNA strand acts as a nuclease, which causes double strand break formation, I guess. Okay. Table 1, DSB induced cell mutation fraction, mu DSB, is the mutation counts n per effective number of sequence CpG sites, mutation site CpG. Mu DSB is that value. The mu and r. Oh, they tell you, yeah, scroll down a bit on the left.
Yeah.
Got it. Okay. The non-DSB induced cell mutation fraction, mu and r, is that next part of that table, next column, and then that is the mutation counts n per effective number of sequenced non-CpG sites. Got it. So the mu DSB is n mutation counts over sequence CpG sites, but the mu and r is the mutation counts n over non-CpG sites. Okay. Mu DSB over mu and r is the DSB induced cell mutation fraction corrected by the non-DSB induced cell mutation fraction. Okay. Induced cell mutation fraction corrected by the non. Okay. So for CPG, we see a pretty high amount of DSB induced cell mutation that is corrected by non-DSB induced cell mutation fraction. But it's much smaller for non-CPG. Okay. Um, all right, yeah, there's a lot going on here. Pretty confusing. So let's see what we can do. Um, was that like your takeaway from this, I guess, also?
Um, and I mean, I kind of understand this part, but to be honest, I just had a tough time understanding everything here,
Hmm.
Frank. I, I was just confused.
Yeah.
After that second paragraph, I was just like, what am I, what am I reading, yo?
Hmm.
Um, but yeah, could you explain this graph, this part again? Because like I guess I kind of get what they're saying here. Because I guess okay, maybe you can correct me if I'm wrong. So essentially, what's going on is that there's these CPGs, these double-stranded breaks happen, which I guess cause like the CPG sites to be methylated, which can lead to mutations.
It seems like that that DSBs can be fixed by homologous recombination HR. Um,
Hmm.
and it could also be fixed by NHEJ.
NHEJ.
Hmm. And they found that um, HR pathway proteins and mismatch repair proteins are co-localized in CPG sites, which are cytosines followed immediately by guanine. Um, and that in the CPG sites, there's just a higher number of methylated cytosine. And this can also mean that there's a higher amount of mutation and due to the spontaneous deamination of the methylated cytosine. And this is like
Okay.
way more likely in single-stranded DNA. And then they kind of go into the HR pathway um, of DSB, and that's what we have here in figure one. Um, so, but it's kind of hard to kind of see what this is kind of showing us. But we start off with, let's see, mismatch repair, chromosomal crossover. So it seems like we have some CPG site and then double strand formation and this. Okay. I guess like the most confusing thing here is like, is this and this like the same thing or are they separate or whatever? But it seems like they're different because, this is the light colored one
Yeah.
and this is a dark. Okay, so let's treat them as different. And let's see. So, okay, this happens, and then we see this, and then you have a stranded invasion. So, like, the darker colored thing can have a strand invade that original lighter gray strand, and that's how it can probably fix things. But yeah, I think that's what I see here. So,
Okay.
um, So it seems like a three prime resection happens, followed by a strand invasion from that darker colored strand into the, the breakage of the lighter colored strand. And then you can have DNA synthesis, ligation, and mismatch repair. And okay,
Hmm.
okay. Um, okay, and then the methylated cytosine. Okay, so yeah, at the CPG sites, there's a lot more of the methylated cytosine, and that can be subject to deamination, um, which I think, because you're talking about, okay, you're talking about GT mismatches. So I think if you get rid of if you deaminate a cytosine, I think
Mm.
you will get a thymine, I think. Let me see.
See, I think it's a urso, right? I think it's a urso.
Oh, okay. Hmm. Oh, well, wait. Well, okay. If you deaminate a methylated cytosine,
Oh yeah, that makes
that
sense.
will
Yeah,
turn into a thymine.
you make a timing. Yeah, that makes
Yeah.
sense.
So, okay, so that's how we can see these GT, like this step here,
Mm-hmm.
like the deamination causes the C with a star is the methylated cytosine,
Mm-hmm.
and we see in the D after the deamination, we just get a TG.
Mm.
So
Okay.
deaminating the methylated cytosine get to your thymine.
Yeah, that makes sense.
So, and that's why this is saying, indirectly causes MMR recruitment to GT mismatches. Okay.
Mm. Okay.
Okay. Now we know that the MMR fixes the GT mismatches, right?
Yeah.
So that's what that will do using HR mediated, or sorry, homologous recombination, right?
Mm-hmm.
And then in experiment one, they took these cells and they they induced DSBs, double strand breaks, and then they measured the effect on the mutations, right?
Mm-hmm.
And so they found in table one that there's a significantly higher mutation fraction in the CPG sites compared to non-CPG sites, right?
Yeah, bigger number.
So we can say that um yes, that the CPG sites have a lot more mutation fractions um during double strand break repair. So yeah, were you saying something?
No, I was just
Yep.
smarting my nose.
Mm-hmm. Good. Yeah. So now it seems like overall, HR repair and MMR can interact um, at CPG sites primarily to, I guess, manage the mutations that come from that deamination.
Mm, yeah, that makes sense. Okay. And
Okay.
then what's the micro NR here? That's just yeah, what is it?
Oh, that just so that's the non so the mu DSB is the DSB induced cell mutation fraction, right? And then the mu NR is the non-DSB induced cell mutation fraction.
Oh, okay, that makes sense. Okay.
So what that also means is that this DSB one is mutation counts n per number of CP of sequence CPG sites, whereas the mu NR is for the non CPG sites.
But I mean, isn't there one for the CPG side here?
Well, that's where the mutation is, but the mu NR term is going to be the mutation rates per non CPG sites. You're still the leftmost columns just shows you where the mutation sites are, like whether on CPG or non-CPG. And
Mm,
but the mu DSB is comparing the number of mutations to the overall number of CPG sites, and the mu NR does the same thing, but it's over the number of non-sequenced, sorry, the sequenced non-CPG sites.
okay. Yeah, got it. Okay.
So that's why so like if you have CPG or if you don't have CPG as your mutation site, your mu and R is pretty similar for both
Mm, yeah.
because that mu and R is in per non-CPG sites. So that's why it's such a small value.
Okay. Sounds good.
Yeah.
Yeah, so if an additional experiment was performed using the cells and protocol described in the passage, what finding would most support the proposed mechanism shown in figure one? Use CPG sites contain 5-ME methylated cytosine prior to TSP induction. That would be harder, right? Because if you have only a few of these, then you wouldn't be able to get this whole process to happen because you probably want you want some mutations to be there or maybe more. So not sure about A.
Let's see which sorry, if an additional experiment was performed using the cells and protocol described in the passage, what finding would most support the proposed mechanism shown in figure one? Figure one mechanism. Okay. Sorry. Okay. Go on.
Yeah, so for A, it just says few CPG sites contain 5-MEC prior to DSP induction. I mean, I think that's the answer I picked last time. But I mean, the more I think about it, maybe that's not right because if you want to have the same mechanism happen, you probably want a decent amount of CPG sites, right? That
Yep.
happen. So
Yeah.
um,
So okay. Good.
MMR localization to CPG sites occurs only during mitosis. I mean, not really sure. The only thing we know is experiment one that they did G1 arrested endothelial cells. But I mean, I like the first part, MMR localization to CPG sites. So it seems that here also we learned that they seem to co-localize in those areas. So, I mean, I'm kind of liking it, but I don't really know.
Yeah. I mean, but they don't really mention much about or anything about mitosis,
Because,
right?
yeah, not really, to be honest.
And it says only, which is like, kind of extreme because it
Because
says yeah.
the only thing they say is re-arrested immediately prior to mitosis. So,
Yeah, yeah. It says, yeah, G1, sorry, G1 as well, right?
Yeah.
G1
G1.
rested, because G0, G1, G2, S, those are all like, interphase, non-mitosis, right?
Yeah.
So, um,
So yeah, I guess B maybe might not be a good answer.
yeah. And then when they say here that the cells were allowed to continue the cell cycle, but were rearrested prior to mitosis, also kind of shows that this
Maybe it's not a good
we're
idea.
talking about some stuff that's happening, not during mitosis.
Gene conversion favors AT alleles during DSP repair. Um, I mean, we see the mismatch happens and they repair it and it does get to AT. So, I mean, maybe, I guess. I mean, that would be kind of helpful to
Yes.
see those repairs. But I mean, the thing is, it's like, even if AT is there, right? I mean, typically AT just um pairs with, yeah, A and T pair with itself. So I'm not sure if that'd be a good way to check if there was a DSP break and all of this stuff happened.
Yeah.
But I don't know. Let me see. D says at least some CPG sites are subject to frequent silent mutations. Um, I don't know. I'm not really sure. I mean, I feel like I don't really know what frequent, I mean, silent mutations, we know silent mutations are mutations that don't really alter like the sequence in any way or that nucleotide. So, but I mean, I'm thinking maybe C, to be honest.
Okay, let's see. So, okay. So C is saying that this gene conversion favors AT alleles. Um, but I don't see anything that really says that it should favor that type of allele, right?
Really?
Whereas, uh
Yeah, I was like, yeah, I still wasn't sure about C, but yeah, I don't know.
so okay, so we know that CPG sites are subject to frequent mutations, right?
hmm
So it should make sense that at least some CPG sites are subject to frequent silent mutations.
But I mean, if even if it's silent mutations, like why would that wouldn't really be a problem, right? I feel like that wouldn't be a problem for repairs or for like all of this sequence to happen, right? Because like it's not gonna really it's not gonna be anything drastically affecting any functions or anything, right?
Sure. But if you were told that something causes lots of mutations, it would make sense that at least some of those mutations would be silent mutation. Like it's not saying that it's making significantly more silent mutations. It's just saying that at least some of these CPG sites have silent mutations,
Mm, okay.
right? And you can
Yeah.
see how it's a lot less extreme by saying at least some,
Yeah.
right?
Yeah.
Whereas, for instance, B says only during mitosis, right?
Mhm.
So like even in the passage, if it said like mitosis,
Mhm.
like over here, rearrested prior to mitosis, let's just say it says rearrested, during mitosis.
Okay.
B would still be wrong because we also have, like, if it was true that this happened during mitosis, we also have stuff happening at G1, right?
Yeah.
So if it's both mitosis and G1, and this says only mitosis, that wouldn't be it, right? And that's even if they said that mitosis was happening. but they say that it's not happening. So yeah, B definitely can't be it. Um, and uh, and yeah, I think the overall because they're asking about the mechanism in figure one. In figure one, we see that this is a repair homologous recombination repair pathway at the CPG sites. And when they talked about it, here, right, they talked about how the uh, methylated cytosine is found with a higher frequency in CPG sites, which causes
Mm-hmm.
an increased rate of mutation, right?
Mm-hmm.
So figure one seems to be talking about, mutations. And if CPG sites, and they're saying that CPG sites have more mutations. So if it had more mutations, it would make sense that at least some of those sites have silent mutations.
Yeah, I see what you're saying. That
Yeah.
makes sense. Doesn't really like specify, but it just
Yeah.
says okay.
Yeah. I mean, I don't know if that's right or not. If it's not right, then that's up to me.
Wait, do you want to check that? We can check actually.
Oh, yeah.
Let's check that.
Oh, you have to like leave the entire thing.
Yeah, that's not too bad.
Okay.
Not too- 30, what was that? 39. Oh, it's C.
Mm,
Hmm,
okay. Damn.
interesting.
Okay, let's see. Figure one shows that in the proposed mechanism, HR-mediated repair of DSP at a CPG site leads to the formation of AT alleles in the template strand. Okay, let's take a closer look at that. This would neither support nor contradict. Okay, I guess they're saying which of, so like I guess D could be a true statement, but they're saying which of these would support the proposed mechanism in figure one, and the mechanism here seems to be, the deamination and all of that stuff. So they're trying to find what would support that, right? So
Yeah.
here, this is saying that, um, let's see, that, yeah, okay, wait. GT mismatches. I'm going to read each of these. So second paragraph, CpG dinucleotides are frequently subject to spontaneous deamination, particularly in ssDNA. The proposed mechanism described in the passage indicates that 3' resection at DSB proximate to a CpG site leads to deamination of the methylated cytosine in the adjacent single strand. Once HR-mediated repair is complete, the subsequent GT mismatch is repaired. If few CPG sites contain methylated cytosine prior to DSP induction, AT mutation could not proceed as proposed. Um, oh my God, I didn't even see that down there. Over here?
ATLU?
Yeah,
Yeah, I mean,
the AT.
I saw that, but I mean, I'm kind of still on the side. Like, even if that is the case, like, let's say there is the conversion and it favors those alleles. Um, like, how would you still be able to tell that the mutation happened? Because yeah, that's
Hmm.
right.
So figure one is just telling us that when you have this process of repair, right?
Yeah.
You take your methylated cytosines, you deaminate them into I'll use blue. So you take your methylated cytosine here, you deaminate it to turn it into a thymine here, and
Okay.
then ultimately this thymine, I guess, gets connected to the or becomes complementary to the guanine here because you have, like whatever, the stranded invasion and stuff, and then you end up with the thymine bound or complementary to the guanine. And we know that can't, well, that shouldn't be possible, but in the last step here, the guanine turns into an adenine.
Right.
So this entire thing is the double strand repair process, and it seems like it ultimately gets rid of a methylated cytosine by deaminating it into a thymine. And here, the thymine and the guanine become complementary to one another. And here, the guanine gets turned into um, an adenine.
Mm, okay. Actually, wait, I think the more I read this too, it kind of makes sense too, because it says, because I keep thinking like, well, A and T always go together. So like, how would you be able to tell this was a result of it? But I mean, it does say like it was performed using the cells and protocol described. So you're kind of, okay. So you're assuming all of this stuff is,
Yeah,
okay.
yeah. Like,
That makes that makes sense. Okay.
so yeah, since the figure kind of looks very complicated and stuff, and like, I didn't look as closely at that.
Hmm.
But when I did, I saw over here that ultimately you get your AT. Uh,
Yep.
and they're really just asking you what finding, like which of the following findings would support the mechanism in figure one. And figure one is showing us that we're going from methylated cytosine to thymine
Hmm.
through deamination, and that the thymine can then be complementary to the guanine initially that wasn't there that was there. And then the last step of the mismatch repair changes the guanine to adenine in order for you to have the AT allele that would be complementary.
Perfect. Okay.
Mhm.
Next one. Yes, I think, oh yeah, this was a silly mistake. When observing chromosome crossover in cell meiosis, students were more likely to have observed what a concurrent event. Crossover, yeah, because that's during prophase. It's not really at metaphase yet. So yeah, spindle formation. Um, yeah, so that makes sense.
So I think B, I mean they, misspelled chromosome, but I think B would be right. Like the first part is right, but it should be at the metaphase plate, right? Where they would
Oh,
line
you're
up,
right.
not
Yeah,
at
yeah, you're
at
right.
the spindle pole, right?
Yeah, yeah,
So um yeah, and the cytoplasmic division would be like the cytokinesis part, right?
yeah.
Um Reformation of the nuclear envelope, I guess, would happen after that as well, after the cytokinesis.
Hmm.
Um
Yeah,
yeah.
yeah, that was just, I didn't even notice the spindle whole thing, but okay, sounds good.
Yeah, they like to do that on. Oh yeah, and this is what we were talking about, I guess, as well. So cytosine to yield thymine involves remove. Oh yeah, because I guess they're just talking about, the deamination part.
Yeah, so just removing amino group. I got that.
Yeah.
Um, whoa. Yeah, this one.
Let's see. Information in the passage and the results of the experiment best support which of the following ideas about. DSP formation and CPG mutational bias. Um, okay.
CPGs. It could be linked by formation of resected 3' ends. No idea what that means. I'm not really sure why C wouldn't be the answer.
Well, it says that the DSPs are induced by like SPO11 in experiment 1, but and also yeah, they didn't mention that CPG sites cause DSPs, right?
It was just that DSPs are typically localized near the CPG sites, right?
The
They're paired by HR.
There's just more mutations at CPG sites.
Then what's the relationship between the CPG and DSPs then? That just
It's in the repair of DSPs apparently because they're repaired by the homologous recombination or the non-homologous end joining. Um, and the HR pathway uses MMRs that co-localize at the CPG sites. So let's see. Information in the passage and results of the experiment best support which of the following ideas about DSP formation and CPG mutational bias. Strong bias against mutation at CPG sites. That's like the opposite of what we see, right?
yeah, there's a lot.
Yeah. And then let's see, they could be linked by formation of resected 3' ends. Let's see, table 1 demonstrates that the frequency of mutation is greatly increased by DSP formation at CPG sites than at non-CPG sites. Furthermore, the passage indicates that DSP repair can occur using HR, the proposed mechanism in figure 1, and information from the second paragraph. Both indicate that DSP repair in HR includes 3' resection at the DSP, and that the methylated cytosines are more likely to be deaminated to yield thymine in single-stranded DNA than in double-stranded DNA. Thus, it is possible that double-strand break repair via HR-mediated pathway induces CG to AT base change in the homologous template strand. So that's saying that they could be linked by the formation of the resected 3' ends. Okay. And then CPG site mutation induces the formation. Yeah, so nothing says that the CPG site induces formation of DSBs, right? That's more so a mechanism, like CPG sites could be more so a mechanism where the DSP repair could happen. And then D says DSP formation
Mm.
induces CPG site methylation, but that's not really the case either.
Like the CPG mutations just happen to be in the DSPs. Like they're just there.
Oh, it's just that um the repair of DSBs by HR um involves, so here I'll clear everything out to make it simpler. So HR, the homologous recombination part of fixing a double strand break, uh,
Yeah.
the proteins for it and the mismatch repair proteins, MMRs, are localized at CPG sites, right? So CPG sites are going to be locations at which proteins involved in repairing double strand breaks rather than causing double strand breaks. So
Okay.
I would, since B is worded pretty strangely, I would do it like this. I would see, okay, A says strong bias against mutation at CPG sites. We know that's the opposite. C says CPG site mutation induces the formation of double strand breaks. Doesn't say that anywhere. We do say here that double strand breaks could be induced by SPO11, right?
Mm.
But aside from that, nothing in the passage states that double strand breaks are caused by CPG site mutations. In fact, it's almost the opposite. The CPG site is where the, repair proteins go or are located, right?
Yeah.
So that's the opposite. And then D says DSB formation induces CPG site methylation. Nothing in the passage says anything like that. They don't talk about what causes CPG site methylation. They just say that there is a lot more methylated cytosine at the CPG sites, but does not connect that to DSB formation. So
Mm.
process of elimination could have been strategy here, yeah.
Okay.
Mhm.
Yeah, to be honest, when I was reading this, when I was taking it,
Yeah.
it was number one, the caffeine,
Yep,
but
yep.
also it was the just honestly by the second paragraph, I was just lost. I felt like I was just lost with the
Yeah,
information.
yeah. And I'm still super confused on this because it's like if this is going this way and this is going this way, it seems like this is a double strand,
Yeah.
but they have this as darker gray one as another one next to it, right? And then they still have this one and this one. So, but I think that has to do with like the double strand break thing. Because like if you have, a double strand here and a double strand here, and you're gonna, or let me make the second one like green, you're gonna have like one of the green ones go up here and form like a double strand with one of the strands, the blue single strands here. I think that's the thing there.
Okay. Absent any mutagenic effects, HR-mediated repair is likely to have the greatest accuracy when the exchange of identical.
Okay, check out this picture I put in the chat.
Okay.
Yeah, so here we have, so this now looks like that thing, right? Because we have like this up here is like in the figure one of that other of the passage. Like this is where you have like the light colored gray, and it's like, I don't know, it was like C or it was like G, C asterisk, C asterisk G. And we have the resection of that. And then this red one here is like the dark gray one from the passage.
Mm-hmm.
And yeah, you can see that there's a double strand break here. And then you have the ends resected. And then you have your strand invasion. the red strand starts to invade the region of the blue strand.
Mm.
Um, oh, and then the blue strand is also entering into the region of the red strand. So that's where you have your yeah. So this is exactly what the
Yeah.
what that um figure was showing us.
Seems very complex. What do you think I should be the biggest takeaway from all of this then?
Oh, I mean, well, it's really just uh passage, information because I don't know if I mean I can look at the content thing. So if we go to the content, BB content, I type in repair. I see repair of DNA, repair during replication, repair of mutations. Let's see if they say like,
It's wrong here. It's weird. Like,
yeah, it really just says repair of mutations, repair during replication. So that's just what you would be expected to understand, I suppose. I don't know if that means
double-stranded breaks.
uh yeah, because the passage, I guess,
Tells you that, I guess.
kind of like tells you, yeah. So
Okay.
yeah.
Sounds good. All right. Yes, I think we have two more questions. Absent any mutagenic effects, HR-mediated repair is likely to have the greatest accuracy when the exchange of identical genetic information occurs between which molecules? Um, I saw it said homologous recombination, so I was maybe thinking like somewhere in meiosis. I couldn't find anything in meiosis. I was like, I don't really know what's going on here.
Oh, okay, let's see. So absent any mutagenic effects, HR mediated repair is likely to have the greatest accuracy when the exchange of identical genetic information occurs between which molecules? Homologous chromosomes during G1, homologous chromosomes during G2, sister chromatids during G1, sister chromatids during G2. Got it. So they mention, okay, so we kind of overlooked this, but they mentioned in the first paragraph that this DNA damage can be repaired using homologous recombination, um, which could involve homologous chromosomes or sister chromatids. Okay. Now, if you're using the homologous chromosomes, that's when you have this, right? As you said in meiosis,
Mm-hmm.
right?
Yep.
But since we know that this is not during meiosis or mitosis, it's like during G1 and stuff. And also the answer choices say G1, G2, right?
Yeah.
So we know it can't be something that's happening during mitosis or meiosis. And they're giving us two like options of how this repair can happen, HR repair can happen, right? So
Yeah.
the if it's between homologous chromosomes, that would have to be during your, meiosis metaphase, right? But
My yeah.
between sister chromatids, right? That could just be between, each chromosome, like within the chromosome.
Mm-hmm.
So we could eliminate A and B because they talk about homologous chromosomes, and that can cause, oh, and then also it's asking about identical exchange of identical genetic information. So we know from homologous recombination that you have crossing over
Yeah.
and thus genetic recombination, right?
Yeah.
So if they're asking about specifically exchange of identical genetic information, it can't be between homologous chromosomes, right? Because that would
Yeah.
be like, from the mother here and from the father here, kind of, right?
Hmm.
So, okay, so we can get rid of A and B, and then sister chromatids, um, you know, and then it's between G1 and G2. And we know that interphase goes like G1, S phase, G2, right?
Yep.
And the S phase, like G1 is like, and G2 is like preparing like the proteins and stuff for your cell division. But
Yeah.
aside from that, um the S phase is the one that causes the DNA replication to happen.
True.
And I think in the passage, maybe on the left, if you go down, they mention, where is it? I saw them mention an S phase.
I said prior to mitosis somewhere.
Okay, let's see. G1. Okay, DSBs in G1. Okay. Continue the cell cycle, rearrested right. Okay, so they're saying that we started at G1 and then it was allowed to go through the cell cycle, but was rearrested right before mitosis. So right before mitosis would be G2 then, right?
Hmm.
So that's why it'll be sister chromatids during G2.
Okay.
Yep.
And that's the last one. Based on the information presented in the passage, which of the following objections would most weaken the conclusion that HR repair induces CPG site mutations? HR repair induces CPG mutations.
Okay.
These are probably
Got it. So if they're trying to weaken the conclusion that HR repair causes CPG site mutations, you could just look at what happens with uh, the double strand break that's using a different mechanism, like uh, the non-homologous end joining, right? Because we are being told that um, that figure one is showing us the HR mediated repair pathway,
Mhm.
and we get to see that it can result in these types of um, mutation at CPG sites. But there's also the non-homologous end joining that can also be a way to repair the double strand breaks. And if that causes mutations, then that could weaken the conclusion that HR repair induces CPG site mutations.
Okay. HR is not required. Okay.
Yeah, let's see the bottom of this passage again on the left. Go up on the left. Okay, so I guess this is just asking um which of the following would weaken the conclusion that HR repair induces CPG site mutations? And the only one that could weaken that conclusion is if you use non-homologous end joining, that could be a way to fix double strand breaks. And that could be another way for the CPG mutations to occur, right?
Hmm.
So so if we do this and we see, a greater amount of CPG site mutations, then
Right.
it could be caused by the NHEJ rather than HR.
Okay. And
Mhm.
then D says HR is not required for CPG site mutation. Um, why is that wrong? HR is not required for CPG site mutation?
Oh yeah, because you could have site mutation from anything, right? From like random mutations from, you know, like mutagens and exogenous agents and things like that. But we're trying to see according to the passage what would weaken the conclusion that HR repair induces CPG site mutations. So we're trying to think, okay, what else could cause these site mutations, right? And it could be the NHEJ mechanism.
Sounds good.
Yeah.
All right, cool.
Mm-hmm.
Yeah, that was really was it.
All right. Awesome.
Hold on. Give me a second. Give me a moment. I'm gonna try to find the phone. So, how much was how much was it today?
Um, should be 156.
Okay. I sent it. Is there any way that you can kind of like just post this as like a listing video?
Oh, yeah, yeah, yeah.